Living cells can tolerate a certain amount of osmosis, but if osmotic conditions change severely, that can have devastating impacts on the cell and can in fact kill the cell. Take a look at the diagram on this slide. If we start over on the right-hand side, we're looking at a depiction of a cell sitting in a solution. This is an isotonic solution. So in other words, the concentration of particles, these green particles that are on the outside of this cell, is the same as the concentration of particles that are on the inside of this cell. Now remember, in our previous diagram involving these green particles, we said that they were not able to cross a membrane using simple diffusion. Perhaps they're too big, perhaps they are too polar, perhaps they have a charge on them. But for whatever reason, they're limited. They're not able to use simple diffusion. Now, in this case, everything is okay. It's okay because there's no gradient of green particle here. There's no gradient across this membrane, so there's nothing that has to be fixed. There's nothing that has to be resolved. We are already at equilibrium. Now the diagram is showing you these blue arrows here, and this is important. Even when we're looking at cells that are sitting in an isotonic environment, I don't want you to think that water isn't moving at all, because it is. Water is constantly moving in and out of cells. It's just that osmosis is not occurring. In other words, we don't see large shifts of water either out of the cell or into the cell. Instead, we see nice equal volumes of water just sort of constantly moving into and out of a cell. It's important to realize that water is always on the move. And in fact, cells have multiple transport processes that move water. Simple diffusion or osmosis is one of the transport processes for water. And we also see what's called facilitated diffusion of water. We'll talk about that in a couple of minutes. This is what we want to see. We want to see cells in an isotonic solution so that we don't have any large shifts in water between the inside and the outside of a cell. Now, if you recall from a previous lecture, we said that organisms that are living on planet Earth are composed of a large percentage of water. We said anywhere from about 60 to 75% water, depending on the stage of growth and development that the organism is at. And we said it's important to note that it's not pure water that living things are made out of, but rather salt water, about 0.9% sodium chloride. We are salty organisms. You could even imagine that this green particle represents sodium chloride, right? Because we said that sodium chloride is going to ionize in a watery environment. It's going to become sodium ions and chloride ions, and because those are charged, they will become hydrated with water molecules. They won't be able to cross the membrane using simple diffusion. Salt is a great example of a molecule that cannot get across a membrane without some assistance. Now, let's take a look at this middle panel. This middle panel shows a cell, again, the same cell from over here, but this time it's sitting in a hypotonic solution. So the environment around the cell has fewer green particles than what are inside the cell. Now we have a gradient. There's a higher concentration of green. Again, let's imagine it's salt. There's a higher concentration of sodium chloride inside the cell than there is outside the cell. We have a gradient. Nature would like this gradient to resolve, to go away, but the particles can't cross the membrane through simple diffusion. What will happen is water will respond. Osmosis will occur. And remember, water travels from the side with the low concentration of particles towards the side with the high concentration in order to dilute it out. So over time, what will happen is this cell will start to swell and eventually it will rupture. We give that rupturing event a name. We call that lysis. So you can imagine this is devastating for a cell. This will kill a cell. And this is something you would want to avoid. Finally, the last panel over to the left shows a cell sitting in a hypertonic solution. There are more particles outside than there are inside. If the green particle is not able to cross the membrane through simple diffusion, water will respond. Osmosis will occur, but this time in the opposite direction. The water will rush out of the cell from the side with the low concentration of particles toward the side with the high concentration of particles, and this cell will collapse. If enough water leaves this cell, the cell will collapse to the point where it will die. So again, in a multicellular organism like you and I, there are physiological systems in place to prevent this from happening. There are systems in place to ensure that osmotic events, large osmotic events, just don't happen because they would be devastating to our cells. For other organisms, such as bacteria or archaea, osmosis events are largely prevented because those cells have a cell wall around them. Bacterial cells, remember, are single-celled organisms, and osmosis could occur at any point, right? If that bacterial cell found itself in a new environment, if the environmental conditions suddenly changed around it, the results could be devastating because of osmosis. So the bacteria have evolved to have a cell wall around them, a wall that will prevent these extreme osmotic events from happening. Let's switch gears now and leave simple diffusion. Let's leave osmosis and talk instead about facilitated diffusion. Facilitated diffusion actually has a lot in common with simple diffusion. It's spontaneous. No energy is required to make it happen. It moves because there is a concentration gradient in place, and the direction of movement will be dependent upon that gradient. It will move particles from the side with a high concentration toward the side with a low concentration, just like we saw before. The only difference is that facilitated diffusion requires a protein. Like the name suggests, the protein will facilitate the movement of the particle across the membrane. Because some particles are too big or too polar or have a charge on them, if a cell has a need for those things to move across the membrane, it will use a protein to get the job done. And that process is what we call facilitated diffusion. Now, these proteins are of two general types. One of them is called a channel protein, and the other is called a carrier protein. And they're both going to participate in facilitated diffusion. They're just going to do it differently. We'll talk about each type of transport protein separately. Let's start with channel proteins. Now, like the name suggests, channel proteins have a channel through the center of them. They have a tunnel that is located right through the center of the protein. And that tunnel is the place where this particle will travel from one side of the membrane through the tunnel to the other side of the membrane. Now, channel proteins are transmembrane proteins. That makes sense. This channel or this tunnel has to allow the particles to get all the way across the membrane. What's interesting about channel proteins is that they have to fold up in a way that allows them to sit in the tail region of the membrane while still allowing things that are large and polar and charged to get through. So that tells you that the inner surface of the channel, what's going to face the particle, has to be hydrophilic, has to be polar. And the outer surface of the channel, the part that's going to face the tail section of the membrane, that has to be hydrophobic and nonpolar. So that means that when a channel protein folds up, the amino acids have to be sort of organized in a way that polar amino acids will line the channel and non-polar amino acids will be on the outside of the channel protein where it interacts with the tail region of the membrane. Channel proteins are very specific. Even though they're open, they're often gated. In other words, something has to happen, some event has to occur to open up the gate so that the channel is exposed and available for transport. Different channels are also sized appropriately so that only particles that are a particular size can get across. A great example of membrane channel proteins are what are called ion channels. Ion channels allow ions to get across the membrane. Remember, ions have a charge on them, and a charge is one of the three things that prevents things from crossing a membrane using simple diffusion. They need help to get across. And the channel protein, the ion channel protein, is going to provide that help. Ion channels usually allow very rapid passage to occur, and that's critical because it turns out that the movement of ions into and out of cells is going to allow a lot of different physiological processes to occur, including things like muscle contraction and nerve cell transmission, signal transmission. Ion channels, like most channels, are highly specific. So in other words, there's a different channel for sodium than there is for potassium, than there is for calcium, and so on. And ion channels are gated. They need some kind of a signal to open up the gate and allow the ion to pass through. We will talk about ion channels when we talk about muscle cell physiology and nerve cell physiology. A good example of a different kind of channel protein is a channel protein called aquaporin. Aquaporin is a channel protein that allows water to cross a membrane using facilitated diffusion. Remember, we said water's kind of unique because water can cross a membrane using simple diffusion. In that process we call osmosis, but water can also cross a membrane using facilitated diffusion. Remember, we said in higher organisms like humans, there are physiological systems in place to try to prevent most of this. to try to prevent large osmosis events from occurring. We still need to be able to move water though, and sometimes quickly. So aquaporin and facilitated diffusion allow that to happen. It allows for a very rapid transfer of water across a membrane in large volumes. And it's found, this aquaporin molecule, in particularly large amounts in kidney cells, in red blood cells or erythrocytes, and it's also quite abundant in plant cells. Now let's take a minute and talk about carrier proteins. Carrier proteins are the second type of protein that is responsible for facilitated diffusion. Now, channel proteins are transmembrane and multipass. And as we said previously, channel proteins have to be lined with hydrophilic polar amino acids. Carrier proteins are a little different. Carrier proteins are transmembrane and also multipass, but they are not going to contain this tunnel or this channel through the middle of them. Instead, they're going to be shaped in a way that allows the solute particle to bind and then cause the protein to change shape. And it's the shape change that will help move the solute particle across the membrane. Just like the channels, carrier proteins are highly specific, which means each carrier is designed to facilitate the movement of one type of particle across the membrane. And this shape change that I mentioned, this process gets a name. It's called the alternating conformation model. So let's talk through what happens with a carrier protein. Carrier proteins will be situated in the membrane so that on one side of the protein there'll be a place for the solute to bind. When the solute interacts with the protein, when it binds on that one side, it's going to cause the protein to change shape or conformation. The shape change will physically carry the particle across the membrane. Now, as it does that, it sort of wraps around this solute, this particle, and it shields it from the tail region of the membrane. Remember, one of the conditions for a solute to need to pass with facilitated diffusion is that it's polar. So these carrier proteins can shield the polar solute, or the charged components of the molecule, from the tail region of the membrane. The shape change that we see in carrier protein physiology is the same as the shape change that we see in enzyme physiology. If you can recall, when a substrate binds to the active site of an enzyme, it causes the enzyme to change shape. It's the shape change that lowers the activation energy of the reaction and increases the speed or the rate of the reaction and allows product to be formed. What's happening with a carrier protein is very similar, although we're not talking about catalysis with a carrier protein. When the solute binds to the carrier protein on one side, the carrier protein will change shape, and it will pull that solute across the membrane and let it go on the other side. Let's take another look at this graph that we looked at when we were discussing simple diffusion. Now let's take a look at the green line, because it turns out while simple diffusion and facilitated diffusion share a lot of things in common, they have different kinetics. Remember, on the x-axis of this graph, we're looking at the concentration gradient getting bigger and bigger and bigger as we go from left to right. On the y-axis, we're looking at the rate of this diffusion. So simple diffusion has a linear shape, and as the concentration gradient gets bigger and bigger and bigger, the rate of passage gets faster and faster and faster. In facilitated diffusion, we have a similar shape to the curve at first. We have a very steep linear shape early on in facilitated diffusion. So as the concentration gradient gets bigger, the rate increases to a point. But then, unlike simple diffusion, the facilitated diffusion line flattens out. If you were to follow it a little bit farther out than this graph shows us, it would reach a plateau. In other words, there will come a point in facilitated diffusion that the speed, the rate of the crossing of the solute across the membrane, it will max out. It will hit a plateau. You don't see that in simple diffusion. The reason that facilitated diffusion hits a maximum rate, a plateau, is because all of the proteins that are being used for the facilitated diffusion are full at the same time. If you're talking about a channel protein, all of the channels are busy at the same time. If you're talking about a carrier protein, all of the carriers are busy at the same time. In other words, you cannot go any faster once all of the proteins are engaged. That's very different from simple diffusion. Simple diffusion will keep getting faster and faster and faster and faster as long as the concentration gradient gets bigger and bigger and bigger and bigger. Now, there's a couple other things to know about carrier proteins, and it involves whether or not they can carry one solute or more than one solute, and what direction they are moving the solute in. So, we give carrier proteins a different name depending on what they're carrying and how they're carrying it. The most common type of carrier protein is what we call a uniport transporter or carrier. This is the kind of carrier that's going to bring a single solute across the membrane in one direction. If you look at the image at the bottom of the slide, this image right here, this represents a uniport carrier protein. This carrier is going to take this solute, called S, from the outside of the cell to the inside of the cell when there's a gradient. So in other words, when the concentration of S is higher over here compared to over here, this carrier protein will move S from the outside of the cell to the inside of the cell until equilibrium is reached. Now compare that to what we would call a coupled carrier protein. Coupled carrier proteins can carry two solutes at the same time. Now there are two different kinds of coupled carriers. There are symport proteins and there are antiport proteins. A symport coupled carrier protein will move two solutes in the same direction. An antiport carrier protein will move two solutes in opposite directions. You can see this in the diagram as well. This protein, shown in purple, represents a symport carrier. it's going to carry both SA, the red one, and SB, the blue one, across the membrane. This carrier will only function when there is a concentration gradient in place, and it has to involve more S red and S blue on the outside compared to the inside, because that's the only direction this carrier can work in. So it's going to carry red and blue particles across the membrane from the outside to the inside when too much of those particles builds up out here. The antiport carrier is going to carry the red particle in and the blue particle out. So this carrier is only going to be functional when you have two different concentration gradients, right? You have more red out here compared to in here, and you have more blue in here compared to out here. It will move red in, down the gradient, and it will move blue out, down the gradient. Let's take a look at how these carrier proteins do this work, because this alternating conformation model is an important thing for us to understand. Now, I like this diagram because it shows the carrier protein in action over time. So this diagram represents a half of a cell. If you were to draw the whole cell, it would be drawn down here. This is the inside of the cell. This represents the outside of the cell. And you can see that the cell has membrane, a bilayer of membrane around it, and a purple carrier protein embedded in the membrane. This one happens to represent a carrier protein called GLUT1, which is a glucose transporter. This would fall into the category of a uniport transport protein. It can only carry glucose, and it can only carry it in one direction. So what's happening in the diagram, if we start here on the left, is this carrier protein normally is open to glucose only on the outside of the cell. So when glucose levels get higher outside the cell compared to inside, glucose can come in and bind to this carrier. When it binds, when it sticks to the protein, it's going to cause the protein to change shape. And that's what they're showing us here. Essentially, it's going to close to the outside and open to the inside. That's going to allow the glucose to come into the cell. That's what they're showing us here. Once the glucose is gone, away from the protein will go back to its original shape, open to the outside. And then we just go around again. The carrier protein normally sits in the membrane, open to the outside of the cell and closed to the inside of the cell. When there's more glucose outside than inside, let's say right after you eat, the glucose can bind to the carrier protein. The binding action of glucose, the fact that the glucose comes in and sticks to the protein, that's going to cause the protein to change shape. It will close to the outside and open to the inside. The glucose will then let go, it'll be released, and this carrier protein that now doesn't have any glucose attached will go back to its original shape. That's the alternating conformation model of carrier proteins. Now, the last kind of membrane transport that we're going to talk about today is active transport. Active transport, like facilitated diffusion, requires a membrane protein. It's a particular kind of membrane protein called a membrane pump. Now, active transport is very different from simple diffusion and facilitated diffusion because active transport requires a source of energy. Membrane pumps are going to move solutes in one direction, either into or out of a cell, but they're moving it in the opposite direction than we see in simple and facilitated diffusion. Active transport involves movement against a concentration gradient. That means movement up the gradient from the side with a low concentration of solute towards the side with a high concentration of solute. This seems very strange. This isn't going to get rid of a gradient. This is going to maintain a gradient. Remember, nature doesn't like gradients. Nature wants gradients to go away. So in order to keep a gradient in place like this, you're going to have to put energy in. So active transport is non-spontaneous. It requires energy from the breakdown of ATP. We call that the hydrolysis of ATP. The outer phosphate group on the ATP molecule will be broken off, and a small amount of energy will be released to fuel this pump. Now, why would a cell want active transport membrane pumps? Well, if you think about things like taking in nutrients or removing wastes, those things might have to occur against a gradient. Think about a cell that is taking in nutrients, for example, from the outside environment. As it takes those nutrients in, more and more and more nutrients are being built up inside the cell. And there'll be fewer and fewer nutrients outside the cell. But the cell needs to take in as many nutrients as it needs. So it might need to function against the gradient. It might need to move nutrients from the side where there's a low concentration toward the inside of the cell where there's a high concentration. The removal of wastes sometimes also has to occur against a gradient. Probably the best known and best understood example of active transport is to maintain ion gradients. As we said a few minutes ago, ions are very important in the functioning of things like muscle tissue and nerve tissue, and it is the maintenance of gradients all the time around the clock that allows for those things to happen when they're needed. So, active transport membrane pumps are used by cells to maintain ion gradients. Probably the best known active transport membrane pump is the sodium-potassium pump. This is a pump that meets our definition of coupled transport, and it's antiport. So it's moving two different ions, sodium and potassium, and it's moving them in the opposite direction. It's going to maintain a gradient. So it's going to maintain a high level of sodium outside a cell compared to the inside of a cell. And it's going to maintain a high level of potassium inside of a cell compared to the outside of a cell. This pump will move 3 sodiums out for every 2 potassiums in every time it breaks apart 1 ATP. This is a very important pump. It can be found in almost all tissues inside the human body.